Bonjour à tous. L'objet de mon
The topic of my speech in the framework of the project is a number of general notions concerning flavonols and their implications in enology, in winemaking. What are flavonols? It's part of the large family of polyphenols, and it is characterized at the chemical level by a skeleton C6, C3, C6, a phenolical core A, and then B with 6 carbon atoms, and then a 3 carbon atom element in the middle. So it's a big family, very big family of plant chemicals. The flavonols are particularly relevant because of their structure in the oxygenated heterocycle, which is what sets it aside from flavanols with an A, which are monomeric molecules, which correspond to tannins as we know them. And they're also set aside from anthocyanins. In terms of the chemical formula and the differences made by the chemical formula of the middle ring. The flavonols of plants, which are very well represented almost in all plants, are found in a glycosylated form. Glycosylation is something that has a role in carbon hydroxide number 3, and it can create glucose, rhamnose, xylose, or galactose. And it can be monomeric or dimeric. In most plants, and it's not the case of vines, what you find is a glycosylation at this level, the 7th carbon atom. As I was saying, flavonols are secondary metabolites of superior plants, which are found in a number of organs. Their primary role, which is generally attributed to them, is a photoprotecting role, protecting plants from ultraviolet radiation, particularly UVB. An important characteristic of this molecule is that they have a role in, they contribute to the color, together with anthocyanins, of flowers and fruit. So they are co-pigmentating the flowers and fruits of plants. Flavonols, from a nutritional standpoint, human nutrition, are particularly bioactive metabolites. They are antioxidants, very powerful ones as part of that metabolism in animal individuals, and they are traps that capture oxygenated radicals, and therefore they have a role in protecting humans from cancer. And that is something that certain pulses do for humans, pulses that are particularly rich in flavonols. Now, let's look at the specific traits of grape flavonols as part of that family. Okay, some terms to understand what we're talking about in grapes. As I mentioned, there are many flavonols which can be differentiated because of the substitutions on the B ring, R1 or R2. And depending on their structure, they can give rise to different flavonols. Quercetin, myricetin, isohamnotin, laricitrin, and syringetin, and camphorol. And they are differentiated. The last three are different from the others because of the fact that the replacing endings are methoxylated. And these are typical anthocyanins found in grapes. Grape flavonols are found in the skins of the berries, but it's also important to highlight that there are flavonols also found in the leaves, which are rich in these principles. It's important to highlight that the flavonols identified in grapes are glycosylated. All of them are glycosylated. On C3, the main forms are glucose, but in the case of grapes, you also find galacturonic acid. Galactose and acetylglucose are a consequence of that. The main flavonols, in terms of prevalence in grapes, are quercetin 3-O-glucoside and quercetin 3-glucuronide. And here are the formulas. This is the glucose, and here's the galactose, and here is the glucuronic acid. In grapevine leaves, there's something specific. Rutin, which is largely represented among plants, is associated to rhamnose, which is very special. This is what's shown here. In the case of red varieties, red grapes, this is seen through the presence of flavonols, such as myricetin, laricitrin, syringetin, which are also glucosylated. These are the 3 substituted flavonols or tri-substituted flavonols. The biosynthesis of flavonols is associated with that of anthocyanins. The white grapes are white because the activity of flavonoid 3,5-hydroxylase is not expressed in them. So if you look at the pathway on the right-hand side, you will see that no malvidin is formed, which is a key in and the flavonols here are not are particularly present. Certain flavonols are particularly present in red grapes. The accumulation of flavonols during their photosynthesis begins with the flowering stage, and there's also a peak during veraison, and throughout the ripening of the grapes, the compounds constantly increase in the berries. The maximum concentration by berry is reached 3 to 4 weeks after veraison. flavanol biosynthesis in general, but that's very well clarified in the case of grapes, is activated by ultraviolet radiation of type B. The cultural or the farming practices that favor the exposure to direct sunlight largely increase the prevalence of flavonols in the berries, while the bunches that are covered by the leaves will tend to develop less of these substances. Concentrations can vary a lot according to the variety, but generally speaking, a study suggests that we have a few dozen milligrams per kilogram of berries of grapes. You find other results in the literature, and there are also posters presented at the forums. We have two posters that provide readings, and the figures suggested there for these concentrations are higher in the posters than here, and that is an indication that in that case it's wild varieties, and therefore that's an indication that the wild varieties are richer in these compounds than what it appears to be the case in this study. Okay, let's take a closer look at the grape flavonols. It's the flavonols, particularly in the berry skin, that is very much exploited during the winemaking process. The flavonols will spread into the wine from the berries, and flavonols, 3 glycosides, are present in the aglycone forms, which are released through hydrolysis. As a consequence of that, over time, during the winemaking process, and particularly maceration and skin contact, it's aglycone quercetin that becomes prevalent in the mix of flavonols in the wine. So it's non-glycosylated quercetin. Generally speaking, the concentration of flavonols in wines will vary depending on the amount of skins used during vinification, and you may well understand that skin contact conditions in terms of duration and temperature have a key role in the concentration, as are the conditions whereby the solids surface during the production of red wines. I will provide, as agreed with the organizers, some information, some pieces of information about the winemaking implications, because these flavonoids, because of their reactions, can have a big impact. The first reaction is hydrolysis. Glycoside flavonols can be hydrolyzed, creating heterocyclic bonds, which are pretty fragile, and that makes them prone to the formation of aglycones. These hydrolyses can be enzymatic, and in this case, glucosidases that can be present are generally more active than the rest of the compounds in forming aglycones, because the glycoside flavonols are particularly are prevailingly glucosides. Also, galactosidases and rhamnosidases are formed, particularly that holds true when there are vine leaves in the process. Lactic acid bacteria also play a glycosidase, perform a glycosidase activity, forming glycosidase and rhamnosidase, and these activities are represented in a study on fermentation, and they're differentiated by yeast strain. Also, enzyme-rich preparations which are used during winemaking, for instance, for settling purposes, will involve glycosidase and glucosidase activities. These activities need to be directed in order to produce more flavonols. Enzyme-rich preparations that are available on the market currently were designed so that the anthocyanase activities are not taking place. So, because of the similar structure of flavonoids between flavonols and anthocyanins particularly, we can imagine that these enzymes, although the glucosidase activities are taking place, this can be directed to the flavonols and not to anthocyanins.
L'autre voie d'hydrolyse est celle de la...
The other hydrolysis possibility is that of a chemical hydrolysis, and that involves the pH of wine. The same applies to molecules of anthocyanins. So again, it is a chemical hydrolysis that can occur and that influences the pH of wine. A second point are oxidative phenomena, which can have an impact on the flavonol component of wine. It has been seen and ascertained that during oxygenation or during the barrel storage, so to say, we had a diminution in flavonols. And this reduction might explain the hypothesis that these molecules are subjected to oxidative phenomena. In addition to that, as I mentioned at the beginning of my contribution, we attribute an antioxidant role to these molecules. They are sort of trap of free radicals. This has been extensively studied, and it has been also seen in food. So these flavonols have this capability of trapping free radicals, and this contributes to antioxidant properties, which is of extreme interest today. So we might imagine and think that even though their concentration is lower than that of tannins, they could have an antioxidant effect. These properties are important. Well, you see, there is also a rebalancing effect in relation to consumption of available oxygen due to the tannins. The third element is the structure itself of quercetin, and I specifically refer to quercetin in this case because we know that the phenolic structure of nucleus B of the flavonol is especially favorable, and we are talking about this ortho-dihydroxy structure. And it is potentially a structure which serves as the basis for the triggering of oxidative phenomena due to the structure itself. The third point I would like to address is about the participation in flavonols to the metal complexes. We have indications in nature which tell us that there are interactions between flavonols and metals. Copper, zinc, iron, due to the special structure, and especially it is chelates, and we have already mentioned them in relation to metals. And then we have ortho-dihydroxy groupings. These structures have not been tested in relation to pH in wine, which is not favorable to the compensation phenomena that have been addressed in literature in more general terms. We might think that at low percentages and low reaction velocities, flavonols can be found as complexes, and this also entails the presence of metals such as iron and copper in that form. There are other possible intermolecular interactions, which is the last topic I would like to dwell upon. And I offer you the example of co-pigmentation. We mentioned co-pigmentation earlier on. Flavonols are substances which are candidate molecules for the formation of co-pigments. This explains the color of flowers and of the skin of fruit. So it is a hydrophobic interaction with a stacking system between an anthocyanin molecule and another molecule, a co-pigment. And as I told you earlier on, flavonols are excellent candidates to restore these types of interactions. They are represented like sandwiches. You see, there is this stacking effect. And indeed, these forms are due to the fact that the nuclei of the flavonoids are almost flat. Furthermore, in literature, it was evidenced based on fermentation studies, not fermentation, sorry, but formation of flavonols, that quercetin is particularly suitable and prone to favoring this co-pigmentation phenomena. So it is suitable for this sort of work in a co-pigmented form with other molecules. Now, I would also like to mention one study that was done in order to investigate whether flavonols also contribute to astringency or astringence. And it is a work that was done in Spain and Portugal. It was evidenced that quercetin 3-O-glucoside interacts with the saliva proteins and also with a model peptide, which are PRPs, proline-rich proteins, which means that phenols can interact with proteins in general. And it was also evidenced using light scattering techniques, light diffusion techniques, that flavonols form aggregates by means of stacking. These aggregations are metastable. This possibly raises a number of questions. What about interaction with proteins? And what about the aptitude of flavonols of interacting with proteins and with the supramolecular complexes that include proteins? Well, in order to answer these questions, we need to refer back to the works I mentioned in relation to astringency and to tests that were made with the molecule of quercetin glycoside in dosages with exceed 200 micrograms per liter. This does not apply in the same way, though, to wine in general. And yet, we can see more details about this aspect, and we'll try and come to a conclusion. With reference to haze and precipitations due to quercetin aglycone, you have, first of all, hydrolysis, formation of aglycone, which is in function of the enzymatic activity. Quercetin aglycone can form hydrophobic compounds and aggregates that are in function of the concentration. Only if you have a certain concentration, you have this phenomenon. And these metastable aggregations lead to a precipitation, which mean more soluble aggregated structures. The cause can also be referred back to over-molecular aggregations with proteins or aminoproteins, and also it can be related to the evolution of anthocyanins, of co-pigments, and you also have oxidoreduction phenomena that indeed can come into the picture. Hence my conclusion. Based on the values I offered and based on data, we definitely can state that we need to know more of flavonols of wine and their future during vinification in order to develop means and tools will make it possible to optimize the richness in flavonols, glycosides, and prevent the formation of quercetin aglycone.